Chapter 7 covers cellular respiration. Energy production within a cell involves many coordinated chemical pathways. Most of these pathways are combinations of oxidation and reduction reactions. Oxidation and reduction always occur in tandem. An oxidation reaction strips an electron from an atom in a single compound, and then the addition of this electron to another compound is called the reduction part of the reaction. Because they always occur together, these pairs of reactions are called oxidation-reduction reactions, or just redox reactions for short. This geothermal energy plant seen here transforms thermal energy from deep underground into electrical energy, which can be easily used. The process that they use for this transformation is just a redox reaction. Removing an electron results in a decreased potential energy in that oxidized compound, so this electron must be shifted into another compound, which is often an electron carrier. Carriers can function like batteries that can be charged and uncharged. The most common carrier that we're going to study is nicotinamide adenine dinucleotide, or just NAD+ for short. It comes from a B vitamin and niacin working together. The oxidized form of this electron carrier is shown on the left, and the reduced form, known as NADH, is shown on the right. The nitrogenous base in NADH has more than one hydrogen ion and 2 more electrons than NADH+. We'll come across a secondary electron carrier known as flavin adenine dinucleotide, or FADH2. It works in a similar fashion, but because its chemical structure, it's not capable of capturing as much energy. So let's look back at our old friend ATP. Adenosine triphosphate has 3 phosphate groups that can be removed by hydrolysis to form ADP, or adenosine diphosphate, or even AMP, adenosine monophosphate. The phosphate groups are negatively charged and favorably interact with water. When the phosphate groups are bonded together, fewer water molecules are able to interact with them, which is energetically unfavorable. For this reason, energy is actually released when ATP is hydrolyzed to form either that ADP or AMP. Once we break up that ATP, the third or even second inorganic phosphate molecule actually just stays present in the cell and floats around. This means that these pieces can actually be linked back together through chemical reactions. Cellular respiration, in the most simple of terms, is the creation of ATP, or the linking of inorganic phosphates, back to AMP or ADP. We label those phosphate groups just so it's easy to keep track of them. The phosphate on the very end of the molecule off to the left is the gamma, beta is in the middle, and alpha is closest to the structure. The reactions that use up ATP are numerous. This very basic image shows you the general concept. ATP donates the phosphorus to a secondary molecule which releases energy. That energy can be used to do physical work, like change the shape of a protein. In phosphorylation reactions, the gamma phosphate of ATP is just attached to the end of the protein, as seen here. We're going to spend this chapter learning about how AMP, or even ADP, is reattached to the free-floating inorganic phosphate molecule and used to create usable energy in the cell. In eukaryotic organisms, oxidative phosphorylation takes place within the mitochondria. In prokaryotes, this process takes place within the plasma membrane. Let's look at cellular respiration in brief before we study all of these reactions far more closely. Step 1 is known as glycolysis, literally sugar splitting. One glucose molecule is split into 2 intermediaries called pyruvates. 2 net ATPs are created in this process. 2 of those NADH electron carriers are also charged. This process takes place in the cytosol. In step 2, oxidative phosphorylation, we move inside of the mitochondria. Those 2 pyruvates are oxidized to become 2 acetyl-CoAs. 2 NADH molecules are charged, and CO2, a waste byproduct, is created. In step 3, also known as the Krebs cycle or the citric acid cycle, those 2 acetyl-CoAs are broken down completely. 2 more ATPs are created, and 6 NADH and 2 FADH2 electron carriers are charged. The last step, step 4, is known as oxidative phosphorylation. Oxidative phosphorylation is broken down into 2 steps because one of them doesn't actually create any energy while the other creates a lot. The electron transport chain uses electron carriers to create a hydrogen gradient in the space between the mitochondrial membranes. The second step of oxidative phosphorylation is known as chemiosmosis. In chemiosmosis, the hydrogen gradient created by the ETC runs through ATP synthase turbine, which creates roughly 28 to 32 ATPs. We're going to look at all of these steps in depth so you can see all the individual reactions that allows us to accomplish all of these tasks. Let's begin our study of the chemical structure. The first step, of course, as you remember, is glycolysis or glycolysis, which is sugar splitting. We're going to see actually how we split that sugar. So the sugar in question is down here, it's glucose. Chemical structure of glucose is C6H12O6. You can actually go through and count all those molecules and see what we're talking about. So here's our C6. Started at the top, I worked my way around counterclockwise. That's actually important in nomenclature. So we have glucose, C6H12O6. And we're going to pop the individual carbons of this off and collect the energy that comes from breaking those bonds. But first we have to give an input of energy. I mentioned that glycolysis is actually, you get 2 net ATP, and these first 5 steps of glycolysis are also known as the energy investment phase because we have to input 2 ATPs in order to eventually get out our 4 ATPs for a net gain of 2. So let's see what's going on. We start with glucose and we add our initial ATP, and we add that initial with the help of hexokinase, these in blue. These are all the enzymes that allow us to do these steps. What that does is it opens up our glucose molecule. So if you count your carbons, 1, 2, 3, 4, 5, 6, you can go through and count your oxygens and your hydrogens. Nothing's changed. We've just added a phosphate group seen here. And this opens up our chemical structure for bonding. So now we have something called glucose 6-phosphate, and that's because this is a glucose, and on the 6th carbon there's a phosphate group. That is acted on by phosphoglucose isomerase. Again, it sounds like a mouthful, but break it down. So we have our phosphoglucose, or glucose with a phosphate on it, and we've made an isomer. ACE just means enzyme. So if you look, everything is exactly the same except for this added structure, which is actually a mirror image of this structure over here in the previous chemical. And this is just a conformational change to help us add energy into this system and create the best product. So again, we're going this time to use phosphofructose kinase because we are now messing with a fructose, and we're just going to add a second phosphate. So now we have fructose 1,6-biphosphate. So we have the fructose sugar because of its conformation, and biphosphate means 2 phosphate groups. And where are they? They're on the carbons that you could label as carbon number 1 and carbon number 6 when you start at the top and walk down the chain. This chemical is fructose 1,6-biphosphate. The next enzyme that comes into play is fructose biphosphate aldose because again, we are going to create our sugar with our 2 individual phosphates, and what we end up with is this intermediary up here and then this intermediary up here. If you see, what we've done is cut this molecule, this previous molecule, we sliced it in half and we added a hydrogen. So now we have 2 different molecules. One of them is this dihydroxyacetone phosphate, and then down here we have a glycerolaldehyde 3-phosphate, also known as G3P. This is going to come up later. You should remember this G3P when we talk about photosynthesis. So we have these 2 molecules. You might be thinking, well, which one goes on? Because they do have different chemical structures, and we know that's important. Well, these arrows look different than all the other arrows that we've had. We've always had one pathway to follow, but in this instance, these arrows tell us that these 2 chemicals can actually freely become one another. They're isomers just in shape. So what happens is when our isomers become this glycerol 3 glyceraldehyde 3-phosphate. That's what we want to work with because the glyceraldehyde 3-phosphate is going to continue on to our next step. So it's up here. The slide reads backwards, drives me nuts. But it's up here. We now have the beginning molecule of what we are going to refer to as the energy output phase. Because now that we've put in 2 ATPs, we are actually going to get an output of 4 ATPs. And we're going to see in this step how we go about charging those batteries. We have our lovely G3P, which, as we have learned, is important in plants who do photosynthesis. So that G3P is going to be acted on by a G3P dehydrogenase. So we are going to dehydrogenate something or take away a hydrogen. Our NAD+, if you remember, that's an electron carrier. And a molecule of inorganic phosphate, the I just means inorganic. A molecule of inorganic phosphate, this is going to take 2 of the hydrogens off of this molecule. It's going to strip it and it's going to add it onto our electron carrier. So we've officially started breaking down our sugar now in order to capture the energy. So our molecule is going to move down now minus some of its hydrogens, and we refer to it as 1,3-biphosphoglycerate. So if we label our carbons 1, 2, 3, we see there are 2 phosphate groups on a glycerol molecule. It's going to be acted on by a phosphoglycerate kinase, and this time note that the ADP is the reactant and ATP is the product. So in this instance, one of those phosphorus, as you can see, or one of those phosphate groups, as you can see, has been stripped away and added to ATP. Our chemical structure is now going to change because there's no longer 2, so it's no longer called biphosphoglycerate. Rather, it's just called 3-phosphoglycerate. Because if you count the carbons, 1, 2, 3, your phosphate group is on your 3rd carbon. It's acted on yet again by phosphoglycerate mutase, and this is just going to mutate the shape of this particular chemical, and now we call it 2-phosphoglycerate because the phosphate group is on the 1, 2, 3 second carbon. Some water is actually going to be released in this step. We are going to get rid of an H2O, and if you count up this chemical structure, you can find exactly where it came from. We have free hydrogens in our system thanks to our NADH battery. We saw that free hydrogen way back over here. Again, we're going to act on our structure with enolase, and it's going to give us PEP, phosphophenylpyruvate, and it's going to change the structure yet again, which is acted on one final time to create pyruvate. And here's where that second ATP comes from. We have ADP over here as a reactant, and we have ATP as a product because this phosphate group has left. Or you only have 2 ATPs coming out of this system, but that's because we started this whole process with 2 G3Ps. So we have to go through this whole thing 2 times. You get the NADH product with the F plus product for each of your G3P molecules, so you end up with 2 pyruvates at the end of this system. And this is the end of glycolysis. Those 2 pyruvates are going to enter into the mitochondria to undergo their oxidation process. The process you see here is glycolysis, and it's happening in the cytosol, not within the mitochondria. Your 2 pyruvates, that's what's going to head into your mitochondria. So your 2 pyruvates are going to head into the mitochondria in order to be oxidized. So here's our pyruvate structure from before, and it's going to be acted on by an acetyl-CoA molecule. That's what's up here, and it actually has a sulfur hydrogen group. What's going to happen is this acetyl-CoA, this enzyme, is going to act on your pyruvate, and it's going to use a battery to help it do that. And remember, these NADH batteries are B3 vitamins plus a little bit of niacin in their structure. It's going to strip electrons, it's going to take them in order to make a charged NADH molecule, and you're actually going to throw out some CO2. This is where the CO2 comes from in your breath. So your pyruvate has now been changed into acetyl-CoA. So you can see the base structure is still the same. This green box is still found right here, but instead you have this sulfur group with this coenzyme group. So the resulting molecule is acetyl-CoA. Remember, you have 2 of these because you started with 2 pyruvates. These end products are now going to enter into the Krebs cycle or the citric acid cycle, 2 names for the same cycle. So here's the Krebs cycle. Lots of reactions yet again, but you can see that it's not nearly as complex as it appears as long as you go through the steps individually. Unfortunately, the enzymes in this instance aren't quite as telling when it comes to their names, but let's look at them anyway. So we start up top and we have our acetyl-CoA. Immediately inside the cell, it is acted on by water, and that SH coenzyme A group actually leaves. So right away, you have citrate, and that's why this process is also known as the citric acid cycle. The guy who invented it, his name was Krebs. So we have the Krebs cycle or the citric acid cycle. I'm okay with you knowing either name. So first we make citrate. This is the chemical structure of citrate. And we turn it into an isomer of itself. So you have isocitrate. An NAD plus electron carrier acts on your isocitrate to charge an NADH battery, and your body releases CO2. After the NADH works on this structure to create the charged electron carrier byproducts, our new chemical structure is known as alpha-ketoglutarate. It's acted on immediately by yet another electron carrier, this time actually with the help of your SH-CoA enzyme. So it gets added back into your structure. So our structure is known yet again as succinyl-CoA. GDP, which is similar to our friend ADP with a slightly different structure, is going to act on this succinyl-CoA, and you end up creating GTP, which is also an energy molecule. Your SH-CoA molecule falls right back off, and you end up with something called succinate. So you have a 4-carbon structure that's going to be acted on yet again by yet another electron carrier. In this instance, it's that FAD. FAD is going to be charged to become FADH2. We have a side molecule that's acting in this instance as well. FADH isn't that great, can't act on its own. Your new molecule, having lost 2 of its hydrogens, is now known as fumarate, which is acted on immediately yet again by H2O to form a chemical called malate. And you can see how the chemical structure has changed if you just look at that nice carbon backbone. Sorry, that is not a rectangle. If you look at that nice carbon backbone to get malate, a 4-carbon structure. Our 4-carbon structure is stripped of even more electrons to charge even more NADHs and release even more hydrogens into the system to become oxaloacetate or oxaloacetic acid. Oxaloacetic acid is what is going to react with that acetyl coenzyme A when the whole system starts all over again for your second pyruvate. So as you can see, you have 1, 2, 3 NADHs that are being charged for 1 pyruvate. But we know there are 2 pyruvates that are going to enter this system. So the citric acid cycle allows us to charge 6 NADHs. We also said that we charged an FADH. But that was 1 FADH2 being charged for only 1 pyruvate. We know we have to double it, so that's 2 FADH2s that have been charged in this system. We also end up charging this little GTP molecule, and that's going to get turned into ATP almost immediately, and that's why we say that we end up with 2 ATPs when you consider that this process goes around twice for your 2 individual pyruvates. What you might have noticed, as I mentioned when we got up here to this oxaloacetate or oxaloacetic acid, that it's going to react again with your next molecule of acetyl-CoA that comes in from the last round, or the next round rather, of glycolysis. You don't have any chemical, or excuse me, you don't have any carbon that's moving on to the next step. As we move into our 4th and final stage of cellular respiration, we are only going to worry about our charged batteries. That's the only thing that's moving forward for us. So let's go there and see what they end up doing. All of those charged batteries end up inside of the mitochondrial matrix. Those charged batteries, NADH and FADH2, are going to feed into these protein structures that are sitting in the inner mitochondrial membrane, and they're going to pump all those free hydrogens that we've been collecting into the inner membrane space. So if you remember every drawing you've ever seen of a mitochondria, It's got that kidney bean shape, and then immediately outside of it is yet another membrane. We are working in structures that are dotting that line. And what we're going to do is we're going to push hydrogens from the inside into the inner membrane space. We're going to use these proton carriers in order to do that for us. Looking at these proton carriers and their names and learning exactly how they function is what you're going to do for part of your flipped class this week. So let's just learn the basics. NADH is going to give up its energy and go back to being FAD plus and a free molecule of hydrogen. That's one of the molecules that's going to be pushed across this membrane. It's going to donate its electron, which will travel as energy through these mobile electron carriers and help essentially power up each of these protein carriers, each of these proton carriers to pump protons from the mitochondrial matrix into the inner membrane space. They work together to create the biggest possible gradient that they can. Finally, all of these electrons are going to flow down out of the last proton pump, having given up all the potential energy they can by falling from a high orbital to a lower orbital, and those electrons will actually be picked up by H2O. We have all of these hydrogens that are eventually going to fall back into this system. Our hydrogens are going to be attached to oxygen molecules that we breathe in to create water. You actually create about 10% of the water that your body needs in any given day. And you create that water by using oxygen as the final electron acceptor in the electron transport chain. So we know that this is the electron transport chain, or ETC for short. These electron carriers are going to donate their little bits of energy, and that's going to allow hydrogens to be pumped into the inner membrane space. Those electrons are pretty amped up, so it takes them a while to fall back down to their lowest state where they don't carry any excess energy. And that when they do that, they end up being picked back up by water. They end up being picked up actually essentially by that oxygen. And we create water. So now we have this huge hydrogen gradient, which is going to have both an incredible positive charge on one side of the membrane and a concentration gradient on one side of the membrane. When we put those two things together, as we've learned in chapter 5, that means that our electrochemical gradient is huge. What do? Well, we run it through something called an ATP synthase. This is that inner membrane space. This is where we have pumped all of those hydrogen molecules. There's a protein structure known as an ATP synthase that actually functions exactly like a turbine. And it's going to open up once this electrochemical gradient has gotten really, really, really high. And it's going to allow all of those hydrogens to flow down through this structure and out the bottom. And in here are actually, you can look up some pictures. I'm actually going to direct you to look to Google for pictures of an ATP synthase. It has these little rotor-like structures, and this whole bottom unit is going to spin just like a turbine. And that spinning, that physical power, is going to be harnessed in order to take ADP and inorganic phosphates that are floating around our cells from having been used. It's going to push them together to create an ATP molecule. And in this particular instance, it's going to create between 28 to 32 ATP molecules. So that's just in this last step. And this last step, if you don't remember from our previous slide, is known as chemiosmosis. So chemiosmosis creates between 28 and 32 ATPs through a process known as oxidative phosphorylation. This is the big moneymaker, but we have to charge all of those batteries in order to get to this step. This is considered the final step in cellular respiration and the most productive. Here's that whole process in one figure. Again, it looks like it's happening backwards, but this is just how our body has set it up essentially. So out here, outside of the mitochondria, this is where your glycolysis is going to happen. You're going to make your products. You'll go from 1 glucose to 2 pyruvates. They enter into the cell where you're going to undergo pyruvate oxidation. Your acetyl coenzyme A will enter into the citric acid cycle, also known as the Krebs cycle, where it will meet up with oxaloacetic acid or oxaloacetate. It flows through, creates lots of charged electron carriers. Those charged electron carriers feed into the electron transport chain, use their energy, combined energy, or to help these nice protein complexes to pump hydrogen protons into the inner membrane space of your mitochondria. When the concentration of hydrogens gets high enough, an ATP synthase molecule will allow all those hydrogens to flow through its rotor-like structure. Its end is going to spin and spin and spin, and it will end up creating about 28 to 32 ATPs. I realize that's kind of seems like a really big gap for ATPs, but you have to remember this is a tiny little structure and this is a tiny little process that's occurring about a few million times a second within your body. And it's not always perfect. We do make mistakes. This happens so quickly. Not every hydrogen might get on its electron carrier. So we say 28 to 32 ATP, accounting for natural error. We think that in a perfect world, it might be 32 ATP that are created, but there's a very good possibility that it may regularly be 28 ATP are created. I only point this out because if you're using external sources, the crash course video for this is awesome, by the way. And the BioFlix animation for this is also quite helpful. Both can be freely found on YouTube, and they're going to mention this 32, and some of the resources you might find will mention this 28. It depends on where you're looking. We're not sure about this. It's too small to be definitive, so we give a range. That ends our whole process of cellular respiration. You went from glycolysis to pyruvate oxidation into the Krebs cycle. The last step is your oxidative phosphorylation, which is broken up into the electron transport chain that doesn't actually create any energy, and then finally oxidative phosphorylation, which creates between 28 and 32 ATPs. That is cellular respiration in a nutshell.